Ketones burn body fat. It's not calorie counting.
We say, what happens if we increase ketones? And so and if I want to increase ketones in a mammal, what do I have to do to insulin? I have to flip it around. And we did this one in rats. We put the animals into ketosis with an incredibly high-fat ketogenic diet. And this is a study looking at the consumption of these very extreme versions of macronutrients in humans. Pure lard, pure carbohydrate, pure protein. And you'll see what insulin is doing, how the insulin response is in each of these instances. Protein's having a modest effect, increasing, and that would depend on the protein, just like it would depend on the type of carbohydrate. But whereas protein has a modest insulin effect, carbohydrate has a substantial insulin effect. And then you can't even see the insulin effect to lard because it's just humming along at zero. That's why when it is a low carb, high fat diet, it's all about keeping insulin low. And you're providing calories to the body, but not sufficient to increase insulin. We pulled the inguinal fat pads, the subcutaneous fat from the rats, and we looked at the mitochondria from the rats. And you'll notice the black bars, the ketogenic diet-fed animals versus the standard chow-fed, the white bars. You'll see that the mitochondria in this subcutaneous white fat from the rats shifted. And you see that the mitochondria are acting, they're much more active. They're using much more oxygen. And then if you'll notice on the bottom with that western blot, it's subtle, it's a little hard to see, but UCP1 started to climb. That it was almost undetectable in the subcutaneous fat of the standard chow-fed rats, and now all of a sudden we start to see it's blossoming, it's growing, the protein is starting to be expressed. And that's likely what's driving this increased rate of respiration in this white fat tissue. Thus, it's shifting the white fat to be more like brown fat.
This diet caused subcutaneous fat mitochondrial uncoupling. And he said this was causing the white fat to act more like brown fat. That is, instead of storing body fat, it is burning body fat.
Now we then went a little further, and this is very recent. We actually grew fat cells in petri dishes, in culture. So we did a cell culture of fat cells to just look, is this happening just at the level of the fat cell? Or is it needing to happen in a whole organism? Do we need the interaction of other tissues? So in this case, we grew fat cells across a dish, and we increased the ketones by simply feeding them beta-hydroxybutyrate. Now, mind you, something that's interesting, in any lab that has ever worked with adipocytes knows, you actually can't grow fat cells, you can't turn them into fat cells without insulin. So you're growing these sort of primordial early versions of cells, the fibroblasts, that can have the potential to turn into all kinds of different cells. And we make them turn into fat cells by giving them insulin. So we have to treat them with insulin for a period of time, then we clear the insulin out, and then we started treating them with ketones to see what would happen. And then the control cells just would stay in the normal control medium. And what we find, the black bars, the adipocytes that were fed ketones, not just the standard culture medium, a substantial increase, a doubling of the rate at which these cells are using oxygen. In other words, we are altogether, when ketones are up, fat cells begin shifting their actions. And they now are starting to use more energy in this uncoupled state, and they're less inclined to store energy.
The results? When ketones are up, fat cells begin shifting. They burn body fat instead of storing it.
So we're shifting the hormones because of ketones, because insulin's low. We're starting to shift the use of energy, how the body's using it. And again, this is important for when we are trying to account for calories.
Annotated and summarized, easy to share with loved ones. The description below the title for this video has these summary points.
In a study at Ben Bikman's BYU lab, they put rats into ketosis, high-fat, low-carb diets. It's all about keeping insulin low. This diet caused subcutaneous fat mitochondrial uncoupling. And he said this was causing the white fat to act more like brown fat. That is, instead of storing body fat, it is burning body fat. In the next study, in a petri dish, they grew fat cells by giving them insulin. To address the question, is this only happening at the level of the fat cell? We turn them into fat cells by giving them insulin. Once they are fat, that is cleaned, They start adding ketones until they are in ketosis. The results? When ketones are up, fat cells begin shifting. They burn body fat instead of storing it. This is important when we consider accounting for calories. If your trainer or doctor suggests that you need to count calories, explain that it is insulin that stores body fat and ketones that burn body fat. It is simply not just the calories. You might consider sharing this video showing Dr. Bikman's work.